In this video tutorial, I am going to show you how to find Cas9 target sites in Drosophila using CRISPR Optimal Target Finder. First, let's grab the sequence of the target region of your gene of interest. Here I will get the sequence of the wingless gene from FlyBase. I will copy the part of the sequence and paste it here. You can select the specific strains of Drosophila melanogaster or other Drosophila species. Here I will use the reference genome of Drosophila melanogaster. Now we got 8 Cas9 target sites in the selected region. Let's check the details by clicking the Evaluate button. In this page, you can check potential off-target sites for each candidate sequence. Here I will select the first target and click the Design Experiment button. Now you need to just purchase these 2 oligos to construct the guide RNA plasmid for targeting the selected sequence. If you like this video, please subscribe my YouTube channel. Thank you for watching.